science dealing with the measurement of the human body to determine differences in individual. Introduced by Alphonse Bertillon in 1900s and spread it to the world in 1940. Composition of Bertillon system have three. Number one is anthropometrical signalment. It is the measurement of the body, head, and limbs. And number two, descriptive signalment. A clear and specific system of description of an individual as well the photography of the male factor. And last one is peculiar mark signalment by indelible marks like scar, mole, tattoos, birthmarks, and etc. Portrait parley, a verbal and picturesque description of person being identified. Such information may be given by witness, relative, or person who is acquainted with the physical features of the person to be identified. For example, mugshot, photography. It is based on Bertillon method. Forensic anthropology. It is the identification of skeleton, study of human remains. It is also a special subfield of physical anthropology. Forensic anthropology and human identification. The role of the forensic anthropologist focuses on applying detailed knowledge of the development, morphology, and variation of the human body as an aid to establishing, confirming, or indeed refuting personal identification. There are four basic biological criteria for the basis of any primary identification. Number one, skeletal or development age. Number two, biological sex. Number three, living stature. Number four, ancestry or racial affiliation. The stages of forensic human identification. Number one, it is human, differentiating human from non-human bones. The anthropologist must be determine whether the bones are human through macroscopic or microscopic evaluating on depending on the elements of the skeleton presented for identification and the degree of fragmentation of the bones. Number two, minimum number of individuals present. Accurate quantification of the number of the individual within an assemblage is the first stage of individuation process. Number three, sex determination. Must know the gender of the bones that examining. Some differences in the skull between male and female. In male, they have large and rugged, prominent and rounded blunt margin skull, rectangular and sloping anteroposteriorly. In female skull, they have small and fragile, not prominent, thinner sharp margins, circular and vertical. Some differences in the pelvis between male and females. Females have wider subpubic angle. Females have sciatic notch less than 90 degree. Females have broad pelvic inlet. Determination of age from bones. Age 0 to 5. Teeth are the best to examine. Age 6 to 25. They have epiphyseal fusion. Age 25 to 40. They have very hard bones. And age 40, they have basically wear and tear on bones. Basic odontology. Application of dentistry in legal proceedings deriving from any evidence that pertains to teeth. Every human body ages in a similar manner. To determine the approximate age, each human has an individual set of teeth, or teeth can be individualized. Teeth is made of enamel. In adult mouth, they have approximately 32 teeth. They have 4 types of teeth. First one is molar, second one is premolar, third one is canine, and the last one is incisors. Teeth differ in size, shape, root types, or root canals. Teeth through the years, for example, the child hoop, it is temporary. Primary teeth expels from milk bud, and to adult, it is permanent. To identification to an individual, it should have postmortem description is generate. Positive identification is compared to antemortem data. Negative identification, a biological profile is generated. And to mass identification, same process is used to individual identification. Organization is crucial. Family is asked to come identify the body and narrow down the full victim. Bite marks, impression from teeth found on skin or items left at the scene, usually outline teeth placement. Where are bite marks found? Not only in the body but the other impressions like food, other items left in the scene. 4 surface that observe the impact. How to analyze bite marks? First, photograph. Cast of impression are taken. Impression traced onto transparency or X-ray. Cast of suspect's teeth are taken. Comparison between suspect cast. Serology refers to a scientific analysis of blood and other human bodily fluids as evidence during criminal investigation. 3 important principal types of cells. Number 1, red cells for oxygen. Number 2, white cells for immune system. And number 3, platelets to produce serum. For presented test for blood, Kastle-Meyer, a bright green color indicates the presence of the blood. It is for visible stains. And number 2, luminol is for large areas where blood is suspected but not visible. If blood is present, it will glow and the color is blue for invisible stains. For confirmatory test, Precimetin test procedure. Once a presumptive test indicates that a stain may be blood, the serologist must confirm that it is human blood. Identifies the presence of protein that are found only in human blood. DNA fingerprinting, also called DNA typing, DNA profiling, genetic fingerprinting, genotyping, or identity testing. When was DNA fingerprinting developed? The technique of DNA fingerprinting was developed in 1984 by British geneticist Alec Jeffreys after he noticed that certain sequence of highly variable DNA, also known as mini-satellites, which do not contribute to the functions of genes, are repeated within genes. Micro and mini satellites galore. DNA satellites were first identified by biologists in the 1960s and come in two types, the micro and mini satellites. The characteristics of micro and mini satellites that makes them useful for identification purposes is that they are highly polymorphic. They can vary significantly in length from person to person. Polymorphic existence of a gene in several allelic forms also variation in specific DNA sequence.
11 get peso call.